Good stuff. Okay, so maybe to start off, we heard earlier about taking a product and improving it through an immunogenicity assessment. With that regard, maybe we can kind of focus on these opportunities for better drugs by this focus on the sequence. And I think kind of when we look at a new product, there's so many things we're thinking about, and it's really critical that those who are engineering are focused on, what issues do we have to face, how can we overcome these. And we had a little bit as well from Professor Choi about the options for looking into those sequences. What always strikes me is that there's, when you look at T-cell responses, there's something like 500 billion different NIMers, which can be a bit scary. And so I think kind of when you've got a sequence and you can focus on some specifics, that it's really helpful to know that you can assess these. So maybe Professor Choi, you can tell us a little bit about the thinking that went into that design and other aspects of immunogenicity that you were needing to think of as you went along. And there's the microphone in the centre of the table.
So in the middle of the talk, the person in the pro-immune, they talked about the over-prediction of the T-cell, the epitopes they're using the in silico method. Now, that's what I actually found it from my calculation as well. So I think the here is like when we talk about the immunogenicity, peptide MHC binding and an actual T-cell response, it's a different thing. But the calculation of the TCR, PMHC tri-complex is just difficult, and it's nearly impossible at the moment because we don't have enough data. So all we can do is actually derive, getting some ideas from the peptide MHC. But it doesn't really mean that it's going to cause the immunogenicity. So, but it's from the perspective of drug development, so it's always good to have the important point is not to miss it. So although it's over predicted, then we just take it and try to minimize as much as possible. Although in fact that predicted part is not actually immunogenic. So fortunately the current the state of the art in peptide MHC binding prediction, it's not really good at. That's just shown in the middle of the talk. It's not really good at predicting T-cell recognizing peptide, but it's really good at the non-binders. So it's a good news for therapeutic developers, but it's a bad news for vaccine developers because it's although you try to make it peptide MHC binding peptide, but sometimes it just doesn't work. So yeah, we are in a good stage. At a good stage of developing safe therapeutics using in silico methods. So, yeah.
Great, thank you very much. And you raised an important point there about this assay sensitivity question. And I think, Daniela, you spoke about this, right? If you have the best assay, you're going to pick up all of the things that are useful, and therefore you'll see true positives and no false negatives. And I think in the field of immunogenicity, we've had this experience with ADA detection where maybe in the early days we weren't detecting all the ADA, so we didn't really know what they were doing. And I feel we're at that point now with these aspects of immunogenicity that I want to see a bit more so that I can decide where to draw a threshold. And maybe if you can speak to your own experience and maybe link in a little bit of agency thinking on this, if you can, that would be lovely. Thank you.
So yeah, it is really an evolving field. I think that I think Malik was mentioning some of the requirements and questions that the agency has asked in the past, and those might not be exactly the same questions that are asked right now. I think that, just like, companies like ProImmune and others have evolved and gotten a lot better at testing, and thinking has also evolved, and the more results we see, then the agency can also sort of evolve in its thinking and say, okay, what is and what is not at risk. So you have to think about it from the perspective of, there had never been an approval of a peptide as generic peptide without clinical studies, right? None. It didn't exist as a path. And so the first thinking was, well, how do we invent a path, and how do we make it, safe enough? And that was sort of the initial thinking. But as we accumulate approvals, as we accumulate products on the market, we are learning, just like we did with the ADA assays, right? We're learning, and the field is learning, the agency is learning, and it's this sort of interaction that is making things evolve. So, yeah, a lot of times I think sponsors press the agency and says, well, we want to know, how much is, where do you want the threshold? They want specific numbers. But I always think that the numbers are actually, it can help you or guide you towards a certain sensitivity, but it also can be a downfall, because it can sort of force you to go into a territory that is not useful, right? If you're all of a sudden looking for mirages or things that don't exist, that is not useful. So this, I think the agency is, it's sometimes, we are blamed of putting guidance very late. And I think it is this learning process. And we learn from whatever you guys submit, and the thinking evolves. But the science is there, and the science progresses. And the idea that I heard all through the talks today from everybody who spoke is, we have a number of tools, but it's not, they're not tools to be applied without thinking. It's not, you do ABC, you're done. Don't think about it. It's quite the opposite. It's like you have this battery of tools. Think about the product that you have. Think about the process by which you make it, and what are the best tools to identify any issues, and how do you construct a strategy? That's where you want to be.
Yeah, super. Thank you very much. Maybe if we go to Julia next and think about your use of the tools and what you see as these issues of sensitivity, the ability to make decisions on your projects with them.
Yeah, it's a good question. So we try to mitigate the risk as soon as possible, because it's like we don't want to have a drug with disadvantage to go to the clinics. So we actually do it before we know the real anti-drug antibody levels. So we need to assess the sequence of the molecule to see if the sequence may have any predicted T-cell epitopes, and we try to de-immunize the sequence so that the molecule will be clean in the end, at least clean from our perspective. Yeah, without any predicted T-cell epitopes.
Yeah, and are you doing that across all products, or are you...
All products,
okay.
Yeah.
So, sometimes you talk about doing things on that risk-based approach, and I, again, would argue that doing what you're doing is the right thing, because even if you're predicting lower risk, if you can remove potential liabilities, then you're much more likely to get the lower risk that you're looking for.
So usually it's a very repeatable process. So we design the sequences, we assess them. We assess them not just for the immunogenicity. You remember, like it's a drug, so the drug should be potent, the drug should be developable. So we need to pay attention to all of these small details when we are doing these repeatable processes of antibody improvement. So we start it's kind of like if you ask me, is it worth to put the money and to put the time? Yes, I will show you tomorrow the data that proves that yes, it is worth to actually to invest in all of these processes, because the patients will get much better drug, and it will have longer lifetime, and it's like half lifetime, and it will better perform in clinics. So, and that's what we are all trying to do, to save patients.
Thank you. So a little teaser already for tomorrow, so well worth coming back. And then maybe if we move to Dr. Balaji there as well. So we've seen like a nice comprehensive view of what can be done to look for the generic peptides. What are your thoughts on, you kind of tied in, aren't you? You've got a sequence, and if it's already got some difficulties, some liabilities, it must be quite challenging to then think about, is it okay to have this, in some cases, relatively high immunogenicity, or are you trying to drive towards, better immunogenicity profiles than the existing products?
Thanks, Tim, for that question. Just let me address Daniela's views on how, agency and the sponsors have evolved. I definitely, second her, statement that, in fact, yes, there were, it was tough, but I think we have quite well moved on. And in fact, on the innate immunogenicity, since almost a year ago, when they said that you focus on the, sameness is enough if you meet certain requirements, the compendial requirements. And I mean, it's a big relief for us. Of course, it's an equally challenging. It's not that easy to prove the sameness. Now, coming back on your question on the challenges, on the assay front, see, we have come across, we went into a situation where a particular impurity was, responding very high in one of the assay. And in fact, by then, that product has gone into the extensive optimization stage, and it has even entered into the commercial batch preparations, and almost, more than 20 batches lying there. And then, the question is, now this became, came as a surprise when we did this. But our chemistry team, the peptide chemistry team, was strongly pushing that based on their prediction, based their mechanism of action, and they are looking at their sequence. They always believed that the response what you have got is not, it is not supposed to be immunogenic. So then we we brainstormed, and then we looked into, see, you cannot just go to an assay which will prove negative, correct? So you need to really look at the sensitivity, and that is where we thought of shifting over to another assay of choice for us, where it came out as negative basically. So yes, at time it challenges. As I said, either if it is so challenging that you go back to the drawing board and rework on it. But here we have reached a stage where, you cannot really afford to get to the drawing board and see how best you can mitigate this risk. So you need to be a little bit, out of the box, in coming up with the solution. So which we were able to achieve.
Yeah, fantastic. I really like the approach you were taking there to say, well, we've been burnt by things, so let's try and anticipate those and other things as we go through each program that we're working on. So just a reminder to the audience, you can still ask questions of the panel at this stage. So does anyone have a question? Maybe there's something you're working on that you would like us to solve for you? Oh, sorry, I hadn't prepared the microphone for it. Is one of these working?
Thank you. Thank you. I just have two questions. In fact, it recommend to how to predict the immunogenicity. Normally, we use in silico and in vitro assay. But for in silico, I think it's very dependent on the also organism you used. Because actually we ever try in our case, we use the same software and the same molecule. But when you choose different algorithm, the result is opposite. So it's very confusing. So I don't know if you have any recommend algorithm is more accurate to predict the more exact results. And the second problem is about in vitro assay, because from now on, the recommend assay is based on PBMC or naive cell-based. I just wondering if it's possible to set up a cell-based assay, like we use cell line like 293T or I think K562 cell line is very good antigen presenting cell model, and also use jacket to replace T cell. Actually, perhaps all these cell lines should be engineering, should be artificial, like to improve the antigen presenting ability. Or like jacket, we know is the TCR is very unique. So perhaps should be do some TCR screening or have more TCR in the jacket population. Like this, I think the reaction will be very pure. Like the signal will be like larger, and I suppose the and this approach will be more sensitive compared to the PBMC system. So that's my question.
So thank you very much for your questions. And as Edmund brings the microphone back. So we've got the two questions here for the panel. Would anyone like to comment on the in silico algorithms and their relative ability to predict and the different outcomes you might get?
So I can start with in silico analysis and in silico assessment. So you know that we are all evolving, as you know. So for example, I assume that 10 years ago the world was in a different spot than now, and I would predict that even in 2 years we will see it in silico analysis with completely different eyes. So I can see already that we are getting to much better predictions, though it's still not perfect. So the first thing that you need to do is to do sequence analysis, proper sequence analysis, and see what amino acids are human, what amino acids are not human, and etc. And what we do internally, in addition, we utilize a few different in silico programs in order to identify the regions. So it's, yeah. But maybe in the future, not maybe, I don't think that it's a maybe. I think in the future it will be much better.
So I can continue with the in silico. So what you bring up has been a big problem for the agency. When I was at the FDA, we would get data from different companies using different algorithms, and it is very difficult for the agency to go back and review all the computational algorithms that are behind. So what we ended up doing is just using the free and available databases and double-checking the results, because it was really a blind box, right? We got results in a very pretty graph, and we had no idea how anybody got to those results. So, we sort of use the available databases that are, available from NIID and freely accessible, and we just double-checked. Because, I completely understand where you're coming from. And different, there's no intent. Just different companies use different algorithms and take into account different things, right? Some take into account registers, some take into account other aspects. Yeah, it's just a whole variety of different parameters or weight. The relative weight of the different parameters is different. So it is a big problem.
Daniela, may I ask you a question?
Yes.
At which stage did agency need this data? Because it's usually...
Okay, so Julia and I are talking about two different stages.
Okay.
She's thinking about when you're probably doing a new discovery, right? So new molecular entity, and I'm thinking generic product, they have an impurity, and there's an analysis of impurity. So it's two different applications, but in her case, she trusts whatever mechanism or algorithm she uses, always uses the same one, and has confidence in her system. In the case of the agency, they get all kinds of results from all different providers of services, and it's the job of the reviewer to understand what is given. And I can tell you that one of the reasons why in silico data is not accepted on its own is because it is so hard to really understand what went into the development of those data. Now, you did ask about in vitro and the idea of using cell lines, which is so tempting, isn't it? It would be so nice and so easy, but it's kind of hard to have. When you're asking, would the patients have T cells to respond, you cannot ask that with a cell line. You can use cell lines in some instances. For example, for looking at impurities, our first screen is usually with a cell line. We don't use PBMCs. We use THP1s and just do a fast screen, and that tells us, is there a problem or not, and sort of that's our first stage. Because it's so high throughput and so easy to do. Or you can use cell lines to answer the question that Julia had before. You have an immune response now, or an innate immune activation, now what? Well, there's cell lines that have specific receptors, and you can see whether some of those receptors are activated. But I don't think you can use T cell lines to look at DC T cell responses. I'm sorry. I don't see that happening. If somebody can, please let me know.
So, about the in silico, because I'm actually developing the immunogenicity risk assessment program as an algorithms, and as the questions that you raised is actually the common complaint actually. Says why there's no simple answer I just give you the sequence and can you predict whether or not it's immunogenic or not? But the answer is not really simple. So in general, we target common alleles. We all have different HLA types, right? So we all have different immune responses. So we just want to. All we can do is statistically, it's likely to be immunogenic in the common population. But the one thing for sure is that the algorithm isn't really accurate for predicting immunogenicity. These days the AI algorithms and the large language models, they can predict some. They claim to predict accurately, but actually they aren't. But what they can do correctly is that the removal of the T cell. The non-binding prediction is pretty accurate. So all we can do, all we can say is we can't precisely say is that the certain sequence is immunogenic, but all we can do is minimize as much as possible against many alleles we have. So the deimmunization is possible, but is the predicting immunogenicity, accurate assessment of the immunogenic for general population is very difficult, so using in silico methods. So, and as the outcome of each method, it's not like a binary. So, oh, well, it's immunogenic or not. It never works in that way. So that all you can have is a kind of a map of epitope probable regions, but that doesn't mean that it's going to be immunogenic. So, yeah, I mean, we are working on it, but we are not at that stage. Yeah.
Yeah, thank you. I think this is a really important point because the tools that are out there are essentially trying to predict whether you get binding to an MHC molecule. So from an in silico perspective, if you were asking how accurate are they at predicting binding to an MHC molecule, you might get a more satisfactory answer and a lot more consistency. There'd be still some false positives, false negatives, and so on, but the accuracy is much better for that question, which is essentially the data set it's trained on. So I think kind of, as you've seen in discussion today, all of these different factors that contribute to immunogenicity, the in silico tools don't include those, so we shouldn't really expect them to be good at predicting like the clinical outcome. One similar thing here as well is even for something like PK, which we sometimes think of as being a lot simpler, when we predict for a phase 1 population PK, we predict for a range still, and it's kind of when we get the phase 1 data, then we refine our predictions for phase 2. If we go into a patient population, we might then refine again for the phase 3. And I think this is probably more practical for immunogenicity as well. Let's try and predict what's going to happen, when we're on the market with thousands of people, because it's highly unlikely we'll be able to do that. Maybe if we return to that second question as well about the in vitro. So we've heard some words on in vitro. I have done a bit of work in the past with the dendritic cell lines when you're kind of looking at specific questions. So I would agree with Daniela, it's rather challenging to, again, kind of predict all the different outcomes. And maybe one of the gaps is on that TCR repertoire that's going to be different in different individuals. I think there are, I guess ProImmune run lots of in vitro assays. There's a lot of value to them. And it's kind of, again, finding what question are you asking. So maybe if we kind of come back to, what are the real positives of in vitro? If you want to reiterate any points that you've made in your talks, or Julia, that you'll make tomorrow, what are the key advantages of using an in vitro, or where shouldn't you use an in vitro? Maybe if we ask the panel that question.
It's simple because it's simple. Currently, in silico is not perfect at all, as we all know, and as you indicated previously. So we do need, even if we get any indication using in silico, we will need we of course we imply in vitro investigations. Yeah, and it depends on the status of this molecule, depends on the urgency of the project, and etc. We have different tools to implement.
Okay, and Dr. Balaji, I think kind of you've shown the value of using these tools.
Absolutely. I think, during my talk also I said, even though, you do extensive in silico analysis, you must prove that and validate that using the in vitro assay. See, in our experience, I have hardly seen in any of those molecules, unfortunately, where the in silico prediction comes out to be either no immunogenic or moderately immunogenic. mean, the no immunogenic situation could have been turned out to be positive in the in vitro. And that is a question always within our company, there are two groups which keeps, the one who strongly promotes the in silico, they have a number of reasons to say that, why it has not. See, I think, I'm not the, too high, expert in this area of in silico. But there is something like non-natural amino acids, which is getting introduced during, any point of the development, which you are not really able to, with the current available tools, as far as I know, I think one of the company is going, pretty well advanced in that area. That's definitely a limitation right now. So that is the reason, one is the agency expectation, the other one is yes, you need to rule out using the in vitro study. So we are adopting that, approach.
Yeah, that's great, thank you. And it's actually reminded me about another in vitro method that's being developed as well with cell lines for the MAPS assay, so the ProPresent type assay. And I think this offers like a few opportunities. So one, it's the monoallelic for the HLA, so you know that it's being presented on that HLA molecule. And this, I think, is where we're probably going to learn a lot more about these non-natural amino acids and what actually gets presented. So not a lot of data in the public domain yet, but certainly an area where the cell lines could be of use to us. Good stuff. Were there any other questions from the audience? We've got one here. We think we'll have to do the running. How long have we got left, Evelyn?
4 minutes.
Just 5 minutes, okay. Cool.
Hi, I have a question about the pre-existing ADA. So we are developing a lot of antibodies. They are VHH-based or SCV-based or FC-engineered. And then it's possible then there may be pre-existing ADA exist in patients. So my question is it necessary to do the early risk assessment of pre-existing ADA for this biotherapeutics? And then is there any guidance or requirements for this? This is number one question. And the second question is about the formats, because we are doing a lot of multi-specific antibodies with different binders, multiple binders. Like, is there related to the risk, the immunogenicity risk of these formats with this? This risk, increased risk, is it possible?
Okay, thank you for the question. So I guess we're not doing so much directly on the ADA measurements. Does any of the panel want to comment on the pre-existing ADA question? I can have a stab at this. Of course, it's good to understand what your risks are for pre-existing ADA, and I think you mentioned the VHH. So there are some reports of specific issue with VHH where there's widespread pre-existing ADA. There's also some fixes that you can do to overcome that by truncating the end of the VHH. So maybe we can follow up afterwards so I can point you in the right direction. And then the other piece is that it's just good practice as you're developing your bioanalytical assays. If you've got a new modality or something that looks quite different than anything that's already out there, it is worthwhile doing that bioanalytical assay development early and assessing this pre-existing ADA question. Because as you develop your assay, you're going to screen a wide range of donors. Now, from a regulatory perspective, you might not need that assay till later. And this is why we would kind of flag up that it's a risk factor that it's something, a novel format, which kind of goes into the other question about the multiple binders. So if I start from a ADA perspective, and then you can kind of go back into the triggering the immune response with the panel. If you've got these kind of multiple binders and it looks like it's very much a different shape, that would already trigger this, it's worth us looking for pre-existing. So again, you don't necessarily need to do like a fully validated assay in order to look for responses. You can do something a lot sort of slimmer package to try and understand if you've got risk. And then you can try and identify where's that risk coming from. You know, is it specific epitopes you can find that you might need to engineer your sequences differently. I will kind of pass to the panel now for, in these multiple binders, there's some complexity going on there. So how do we use the current assays and what do we think about assessing the risk of these?
I can comment on the stage of preclinical assessment that any modification to the sequence you're doing, any new format that you're introducing in vitro, it can potentially increase immunogenicity risk. Will it be implied later on? Maybe, maybe not, but you need to assess all of these modifications in vitro before progressing, because at least you will mitigate the risk.
Okay, any other comments on that one?
Well, actually, it's not about the multi-binder, but it's just about the pre-existing ones. I think the pro-immunes that they worked on is the Cas9 thing, and it's in 2019 the researchers, they found that Cas9 is from the bacteria, so they are, those who are infected by stop aureus, the strains, then so they tend to have the pre-existing ADA against the SA Cas9 as SB Cas9, and I think the risk there is, is that if you develop the drug, and as if it has like a pre-existing immunity, then it can have they reduce the efficacy. But the worst case is actually one of the antibodies, they change the isotype switching, and it becomes IgE, and so it gives allergy. So I think at the moment is the allergy prediction is I think Daniela, yes. And she pointed out that the in silico method of like B-cell epitope prediction, because it's basically the antibodies. But at the moment, B-cell epitope prediction tools are not really reliable. Compared to the T-cell epitopes. We keep developing, but I have to say nothing really works. So we are quite behind the predicting allergy and its allergenicity and all those things. So I think the pre-existing ADA, I think if it causes, if there is a chance to induce allergy, then probably you have to get rid of it. But do it with in silico, I pretty doubt. I doubt it's possible to do it right now, but you can still try. Yeah.
Great stuff. So on that question, sorry, Nick, yeah. So.
Yeah, one comment on that with pre-existing antibody responses is that one thing you can think about, and it may not be relevant in all cases, is you can also look at memory T-cell responses, of course, in this context. Now, the epitopes for the antibodies will not be necessarily the same, in the same region as where the T-cell epitopes are coming from. But if your memory response comes from a certain element of a molecule that, maybe your patient has had that looks similar to the molecule you're now designing, then, it's quite likely that whatever caused the, pre-existing antibody response is sort of co-packaged with some T-cell epitopes where you have memory for those epitopes as well. So the proliferation assays can be easily further phenotyped into distinguishing naive from memory responses. So one of the controls we often run in these, or I think run in all of the T-cell assays, is PPD, tuberculin PPD. And you can clearly see that that's a memory response as opposed to the KLH response, which is clearly naive. So if you phenotype those responses down into memory and naive phenotypes, you can clearly see that every single time. So again, that may help you to, pick apart a little bit more the specific problem that you may be facing and potentially answer the question of where does your pre-existing ADA even come from.
Great, thank you, Nick. And I'm going to get this back to the panel. So we're about to close, so I think it would be really nice to hear the panel's thoughts on, given what we aren't able to do at the minute, apart from just predicting immunogenicity, what do you think is the next key development, either technologically or behaviourally, we need to do differently to get closer to true prediction of immunogenicity? I should have primed them for this, shouldn't I?
Should I give
Yeah, please, go ahead, thank you.
Well, I mean, it's basically it's a free comment, right? Yeah, well, this is my like long-standing questions. There's the immunogenicity. I mean, I'm actually you can call me as a computational biologist. So I'm not really doing in vitro, in vivo, or whatever. I just try to do everything in computer, but although I believe that the power of computer and the recent the development of the AI the technologies, but I think we still don't know very much about the immunogenicity. So it's, and the problems in it's the industry they are interested as like two extremes. It's like they want to make it general drug, which works for everyone. Or the other extreme is like they want to make it personal. But I think that there's something missing in the middle. So it's like some mesoscopic scale. So for example, so it's like neoantigen development. So for example, different ethnic groups, they have different HLA types. So they might respond in a different way, but nobody really cares about it. So I think maybe it doesn't make money. So it's not like the personalized, so it's not like the general enough. But I think as an academic, so I think that there's some niche needs. For example, as a Korean, so I'm a Korean, and we have a very specific type of HLA class 1. And so if you find the tumor, cancer patients, and so if you sequence them, then they have, Korean-specific HLA types, which are not really registered much in IDB, which means that it's highly driven by, well, is it politically correct way to say it about the Caucasians? So, but we Asians have different the HLA types, but no one really cares about it at the moment. I think the immunogenicity issue is the same. So maybe it works in a FDA and US there are lots of the ethnic groups, but maybe Asian-driven world and different Latino group world, maybe they respond differently. But I think as an academic question, that's a really great question to see immunogenicity works in different ethnic groups. But yeah.
So to make immunogenicity assessment like a truly global endeavour for humanity as opposed to just smaller markets. Thank you, Daniela.
Well, there's a couple of things that I would like to see. I think that, okay, I'm going to just name three. First of all, we've focused on antibodies because antibodies are easy to measure. But antibodies are by no means the be and end all of the immune system. It's just one aspect, right, of an immune response. That's number one. It links to number two, which is we've gotten pretty good at measuring the incidence of antibodies. We have not gotten good at all at understanding or being able to predict what the clinical consequences are. And I think that that is possible, and we need to work in that direction, because developing antibodies is not an adverse event. And if we can get better at understanding what the clinical implications are a lot of products would not die, and in the preclinical stage, they could go forward because we would understand that the antibodies would not have an adverse event, and I think that would be important. And the third thing that I think we're really lagging in is we still are nowhere close to being able to do good prediction in hypersensitivity. And I think that is really an important need. These are, usually a lot of times it's an emergency situation and it can have devastating effects for the patients. So we really need to get better at understanding what can drive hypersensitivity and so that we can mitigate that risk.
Super, thank you very much. And Julia?
Yeah, I will be quick, but it's like for us, and I come from the industry, it's all about speed. So if we would be able to have assays that can indicate immunogenicity, demonstrate or not demonstrate immunogenicity faster, it will be much better, because to wait a few good months to get the results while we are still assessing the candidates and etc., it's really painful, I have to say. So yeah, if we can kind of as whole scientific world can make it faster, it would be really great.
Super. I think we're nearly at the end, so we'll have the last word. Dr. Balaji, what would you like to see? We'll come back to Nick. And Nick's got an answer to doing things quicker, by the way, so I'm sure we'll hear about that at some point, but let's take Dr. Balaji.
I will not take much time. I agree with Julia. Again, what she said is right. This is not just the time, which consumes, for these assays. And for a generic drug company, definitely the cost matters. So, Daniela is now out of agency. I don't know whether I would have asked this question. Now I take the liberty of asking this question. Many a times, the management asks us, when you are spending so much money on the in silico in vitro, why don't you do it in the clinical study? Anyway, we are doing some clinical studies and do the ADA analysis. Would that be, sufficient enough?
Well, for the FDA at least, and I'm not talking about any other regulatory agency, but just the FDA, you can either be a 505(b)(2) as a follow-on or a generic. Generics do not contemplate clinical studies. The advantage of having a generic is, of course, you get all of the indications and the interchangeability at the pharmacy. So it's an economic, I guess it's an economic decision for the companies. What they want and what their strategic investment is. This is not about science. It's about economics. On the other hand, I would say, let me put it to you the other way around. You have a product that has an incidence of immunogenicity, let's say 1%. What difference do you think would be acceptable, right? So, and how many patients do you need to prove that, right? So if you had a product that has an incidence of 1%, and I think I may be presenting some of this tomorrow, but if you want to discard a tenfold change, it may be enough to do 200 patients. But if you want to discard just a 50% increase in immunogenicity, that's 1,000 patients. So you need to do the math and really figure out whether it is cheaper. But at the FDA, we're forbidden to think about the money. It's we are literally told you cannot think about the money, either the money that the company will make or the money that they have to invest. That's not your problem. Your problem is to get a safe and effective product. So we tend not to think about it. And sometimes, it is a lot of costs for the companies up front. And we're not supposed to think about it, but it is difficult not to, because we don't like to see products fail because of the hurdles.
Thanks.
Okay, so did you want a word there, Nick, before we close the panel?
Yeah, just to comment on the assay speed. So it's something that we've been thinking about and been trying to focus on a lot. And I think there are just a few variables that are a bit fundamental. One, with the peptide style assays, whether this is MHC peptide binding or T-cell assays, it's the time to synthesize the peptides. And the problem you typically have is that you need to synthesize, or a lot of projects then involve a lot of peptides that need to be synthesized at high purity. And so the issue is that peptide synthesis is a sort of statistical process. Some peptides will always fail initial synthesis and purification, so you have to try again and again and so on. So we have to plan in a minimum amount of time to get all the peptides ready for the study. So, but the sponsor can sort of respond to that in a sense by pre-planning it, pre-ordering peptides, and I think that's what some sponsors certainly have done. And then later they pick up which peptides they really want to take forward into specific analyses. So that can certainly, there's a cost implication, but it can certainly help. And the other issue where we see delays, where we actually test drug products and comparators, is sponsors are super excited about being very fast, trying to test their product. The problem then comes when they need to supply the drug and the RLD to us. That's where, we try to be very proactive in helping the sponsor think about where is this thing going to come from inside your company and arrive at ours, so we can actually test it. So we're ready often, and we're just waiting for the drug. In some cases, we've had people turn up in person from South America to deliver the drug to us so we can put in the assay, because the shipping is too complicated. But yeah, I mean, it's something that we can sort of talk through and make sure that there's no disappointment along those lines. So these are the sort of two factors that are maybe sometimes not so well understood. On our side, we are very quick if we need to be. Yeah.
Thank you, Nick. And I will just give you a testament here that I certainly had an example once where we needed data quickly, and although we wanted 40 peptides worth of data, it was helpful to have the 35 peptides worth of data, the ones that synthesized more easily. And so ProImmune were able to deliver that in a shortened time frame, because occasionally, sometimes timelines speed up. Most of the time they slow down anyway, but occasionally they speed up. And certainly I've enjoyed working with ProImmune to be able to be flexible in delivering data as needed. So I think we're going to close the panel here. Thank you ever so much for the panellists. And before you leave the stage, so we'll see Julia tomorrow. Daniela, we'll see you again tomorrow. So there's lots of exciting things to go on tomorrow as well. And if any of this is triggering thoughts as to what questions do you have, do feel free to ask any of us your questions, the challenges you're facing, or just if you're curious about the science. We also love talking about that as well. So, Edmund, are you closing us for today? Yeah, so thank you very much, everyone.